Welcome everyone to this week's edition of the Extracellular Vesicle Club. We have a guest moderator today, and we have an exciting presentation that's coming to us from the University of California, Davis. This is a continuation of our mini-series on single particle analysis. And so we're going to be hearing from a lab that's one of the leading labs in the world in this area with multiple techniques. So I'm very excited to hear today's presentation. And so now our guest moderator is Fabrice Lucien. Fabrice, could you just briefly introduce yourself and then our speaker? And thanks so much for moderating today.
Yeah, thanks, Ken. So everyone, hello everyone. I'm Fabrice Lucien, I'm assistant professor of urology at Mayo Clinic. And obviously, I'm pleased to moderate today's journal club. So I will introduce Dr. Randy Carney and his PhD candidate, Rachel Mizenko. And after Rachel will present her work on EVs. And if you have any comments or questions, please just use the chat box. And at the end of the talk, I will either read the question or say your name so you can ask directly your question. So Dr. Randy Carney is assistant professor of biomedical engineering at UC Davis in California. And his work has been focusing on developing new technology for cancer diagnosis using EVs. And I believe that a lot of us are familiar with his work because he presented, I think, at the EV Journal Club last year on the potential of nanoplasmonic platform combined with Raman spectroscopy to characterize circulating EVs from biofluid. So today we have, I'm pleased to introduce Miss Rachel Mizenko, who's PhD candidate in Dr. Carney's lab. And she will present a recent work published in Journal of Nanobiotechnology, which provides actually provocative findings on the expression of tetraspanins on the surface of extra vesicles. So Rachel, floor is yours.
Great. Thank you for that introduction. Nice to see everybody this morning. As I said, my name is Rachel. I'm a PhD candidate. It's really nice to be invited to this since we are so interested in this single particle characterization. So as he said, today I will be introducing my most recent article that was published in the Journal of Nanobiotechnology. And it is titled Tetraspanins are unevenly distributed across single extracellular vesicles and bias sensitivity to multiplexed cancer biomarkers. So today I'll be starting by giving some background, first on ovarian cancer diagnostics and why we need new ones. And then I'll get into the use of tetraspanins in EV diagnostics and elsewhere so everybody can understand why this is important to them. After that, I will get into my results, first on tetraspanin heterogeneity, and then on non-specific capture, specifically by our biotinylation method. At the end, I'll give you a summary of our results and some key takeaways from the paper. If you are familiar with the paper, it is a fairly long one. So there are a couple things that I am not planning on going over, such as interferometric imaging. Please feel free to ask questions about this if you are interested at the end. So with that, I'll get into my background. So about 1 in 78 women in the United States will be diagnosed with ovarian cancer in their lifetime. And unfortunately, about 2/3 of these individuals will die of the disease within 5 years of diagnosis. Obviously, this is a really, really poor prognosis, and it mainly can be blamed on the low sensitivity and specificity of the current clinical standard for diagnosis. This is the CA-125 test, which tests the levels of the CA-125 antigen in the bloodstream. However, the people that have early stage ovarian cancer, stage 1 and stage 2, only about half of these individuals have high levels of the CA-125, excuse me. This means that the majority of individuals will be diagnosed during late stages, stage 3 and stage 4, and this is where the survivability of the disease drops below 40%. So obviously this is not great. Second, the CA-125 test is also not very specific. So of all individuals that test with high CA-125 levels, only about 20% of those individuals actually have ovarian cancer. And this is because the CA-125 antigen levels, they sort of cycle with different things that are benign diseases. So this means that there's going to be a longer wait until people can actually treat ovarian cancer, since we need to confirm by biopsy that that is what the CA-125 level is indicating. So together, this low sensitivity and specificity of this current diagnostic tool shows that we do need new diagnostic platforms so that we can detect ovarian cancer at these earlier stages. This is where EVs come in. EVs are sort of a great replacement for that CA125 test for two main reasons. First, we know that EVs carry proteins from the cells that produce them. So specifically, ovarian cancer EVs will carry proteins such as EpCAM, CD24, and HER2, which are all known to be associated with ovarian cancer. Second, we're able to find these ovarian cancer EVs in circulation in the bloodstream. So we should be able to keep this non-invasive blood test to test hopefully for higher sensitivity of this ovarian cancer. So because of this, a bunch of different groups are testing different ways to use EVs as this target diagnostic tool. And there are a bunch of different things that people are doing, things like ELISAs, bead-based immunocapture, and different microfluidic sort of lab-on-a-chip systems, which is very, very exciting. However, there seems to be a sort of commonality between all of these different platforms, and that is that these labs almost always start with primary capture of their EVs by a single tetraspanin. So people in the EV field are probably very familiar with these tetraspanins, CD9, CD63, and CD81, because they're known as these EV-associated markers because they are enriched in small EVs compared to whole cell lysates. However, more and more work is showing that these tetraspanins are not homogeneously expressed across all EVs in a population, even from a single cell line. So a couple of these works show that there are these subpopulations of this tetraspanin expression. This means that when we capture by a single tetraspanin, we're only getting a fraction of the total EV population. Currently, there has not been any work showing whether or not we have these same subpopulations in ovarian cancer cells or EVs from these cells, or if there are also subpopulations of these tetraspanins that multiplex specifically with these ovarian cancer markers that we're interested in looking for. However, this use of the single tetraspanins is not something that is unique to the diagnostic portion of the EV field. And in fact, single tetraspanins are used in all kinds of different studies in the field. Here I just show two that used transfection of a parent cell line to fluorescently label CD63 on the surface of EVs. These studies are really great. It tells us a lot about how EVs work and where they go when they do have this specific tetraspanin. However, I think a lot of different people, when they read this, they think of this as indicative of the entire population of all of our EVs. However, if it is this subpopulation, we really don't know if there are functional differences between these subpopulations. So do EVs that carry CD63 always have the same function as those that do not carry it and carry CD9, for example? So this really is something that affects all portions of the EV field. So back to my ovarian cancer sort of diagnostic portion. Like I said, there's been no study currently that is showing these subpopulations on SCOV3 derived EVs or on ovarian cancer derived EVs, or if there's any multiplexing biases with these tetraspanins and ovarian cancer markers that we're interested in. This led us to ask two main questions. The first, what is the tetraspanin expression profile of EVs from ovarian cancer cells? And second, can EV capture by a single tetraspanin affect identification of ovarian cancer markers? So this would tell us, number one, are we getting these subpopulations? And number two, are they of like clinical relevance? So to answer this, we began culturing ovarian cancer cells. We chose SCOV3 as this is an epithelial ovarian cancer cell line. And we cultured these in a bioreactor and isolated the EVs from this bioreactor by differential ultracentrifugation. Here is the general sort of characterization tools that most labs are going to use. We started with transmission electron microscopy, confirming that the EVs that we isolated were vesicular in structure. Moved on to nanoparticle tracking analysis, confirming that we had isolated small EVs that were smaller than about 200 nanometers on average. And finally, we looked at Western blot to confirm that we did get these different tetraspanins that are indicative of EVs in our isolate. So this was great. We confirmed that we had EVs, and this is where a lot of labs stop because you do get that confirmation. However, because our lab is specifically interested in these subpopulations and this heterogeneity and these single particle analysis techniques, we go one step further with this characterization and ask, what are we missing with this bulk analysis? So with this Western blot, we see that we have these tetraspanins, but are there subpopulations? Do they have clinical relevance? So this brings us to our first question, answering this first question of what is the tetraspanin expression profile of EVs from ovarian cancer cells? So do we have those same subpopulations that people have found on EVs from different biofluid sources? So to answer this, we needed a way to look at single EV protein expression. We decided to do this using the ExoView system, which combines immunocapture with immunofluorescence. So this is a fairly new machine in the field. So if you're unfamiliar with it, I like to think of it as a combination of sandwich ELISA with microscopy on top. So with this technique, you have a chip that has a microarray of antibody capture spots, specifically here for these 3 separate tetraspanins. You can incubate EVs on top of this chip, and they will be captured by these individual capture spots. After this, you wash away any of the unbound EVs or particles, and then you secondarily fluorescently label with antibodies. Specifically, we used fluorescent antibodies for those same 3 tetraspanins, as this would allow us to know at least 2 tetraspanins on single EVs. So what I mean by that is you have the tetraspanin that the EV was captured by that you know, and the fluorescent antibody that tells you which tetraspanin it is labeled by. So here at right is some of the raw data, and you can see at that top right corner there, a white single spot, which indicates that we have a single EV with all 3 of these fluorescent antibodies, indicating all 3 tetraspanins were labeled. So this is great. It gives us a way to look at these single particles and their individual expression of these different tetraspanins. However, the first thing that we needed to do was confirm that we were in fact looking at these single EVs. So what I mean by this is each of these individual spots is diffraction limited. So the actual signal of fluorescence is much larger than the actual EV itself. So we need to confirm that these airy disks of this fluorescent intensity are completely separated. This would allow us to say that the number captured actually correlates with the concentration of these different subpopulations. If we didn't do this, we would see this lower image where we get signal crossover from EVs that are bound nearby. So the software itself identifies if things are single EVs, and when we have this signal crossover, the software cannot identify single EVs and instead thinks that multiple EVs are going to be a single particle. This means that this is no longer single EV analysis, and the number that it produces is not going to correlate with the actual concentration of those different subpopulations. So to confirm that we were actually looking at single EVs and figure out sort of what concentration of EVs actually represented this single EV regime, we did a dilution series of our SCOV-3 EVs on top of these chips and counted the number of EVs that were captured on a single capture spot. Here we found that over a concentration of about 5 × 10⁸ particles per milliliter when measured by NTA, we started no longer seeing a double in the number of EVs that were captured when we increased the incubation concentration by doubling it. So this could have indicated one of 2 different things. First, it could be that the machine, the software, was analyzing multiple EVs as a single EV. Or it could indicate that there were some weird binding kinetics going on, since this does require binding of the EVs to the chip surface. So to determine which of those 2 things was happening, we then started looking at the average fluorescent intensity of each of these points. So if we were looking at single EVs, no matter how many EVs are bound, we should see that the average fluorescent intensity remains the same, because we're looking at the average of the population. However, if we're seeing this crossover of signal where the machine thinks multiple EVs is actually a single particle, we would see an increase in the average fluorescent intensity since we're combining the nearby fluorescence from 2 particles. With this, we found that over a concentration of 2.5 × 10⁸ particles per milliliter when measured by NTA, we started to get this increase in the fluorescent intensity of each of our points. This indicated that we needed to use this concentration from now on to get these single particles. This concentration correlated to about 2,500 particles in a single channel on a single spot. So we tried to use this as sort of our limit to say that, yes, we are in fact looking at single EVs from now on. So now that we had this value, we started looking at the actual protein expression profile of these EVs that were captured. So here is the sort of data that you get from this instrument. I'm going to go through this a little bit slowly because I know it is rather dense data. On the x-axis here, you get the capture spot. So which of these tetraspanins that the EV was captured by. So these first three columns were all captured by CD9. And on the y-axis, we get which of the fluorescent antibodies the EV was detected by. So here in this very tall green column, you see that this is captured by CD9 and detected by CD81, which means we know that there's multiplexing of those two tetraspanins on single EVs very frequently in this population. So had we seen homogeneous expression of these different tetraspanins, we would have expected to see that on each of these different capture spots, we saw the same amount of EVs for a single detection antibody. So we would have seen the same number of CD81 detected antibodies or detected EVs on each of these three capture spots. However, as you can see, we actually get significant difference based on which capture spot. So we see very high detection of CD81 on the CD9 capture spot, but not the other two capture spots. And this is the same for the other two tetraspanins as well. This indicates that we are getting these subpopulations, which was really exciting. So before we had any big takeaways from this, we started thinking, well, maybe this is something that was an artifact of this single isolation, or it could be something that is transient. So we wanted to see, are these subpopulations of tetraspanin expression sort of constant over time for these SCOV3 EVs? So we did 6 consecutive isolations from that bioreactor of our SCOV3 EVs and compared them to see if we were getting this consistent profile. And in fact, we did see that this tetraspanin expression profile did seem to stay the same. We always got this relatively low detection by CD9 and a high detection by CD81, which was really exciting. So with this, we had confirmed that we are seeing these subpopulations that other labs have seen from different biofluid sources on their own EVs, which was great. So our next step was trying to figure out, is this something that is unique to SCOV3 EVs, or is this something that is common among all biofluid sources? So do all EVs have these exact subpopulations of tetraspanin expression, or is this something that changes based on where you get your EVs from? So to sort of identify this, we compared our SCOV3 EVs to EVs from 2 different clinically relevant sources. So we used serum-derived EVs and placental mesenchymal stem cell EVs for their diagnostic and therapeutic relevance. With these, we found that there actually was a significant difference in their tetraspanin expression profiles. So here you can see at top right, with these serum EVs, there was very high detection of CD9 on the CD9 and CD63 capture spots, which we had not seen on our SCOV3 EVs. For placental mesenchymal stem cell EVs, we found that there was very high detection of CD9 in comparison to SCOV3, especially on the CD81 capture spot, which was different from what we saw with our serum EVs. So this was really exciting. It indicated that all of these different EV sources had very unique expression of these tetraspanins, and that it was consistent on these different EV sources over time. So with this, we were hoping to get these same results on an orthogonal technique and compare them and recapitulate this data. So we decided to use flow cytometry, since that is the most common way to look at single EV protein expression. So here I present the same ExoView data, instead looking at on a total chip, how many or what fraction of the EVs were detected by a given fluorescent antibody, just to simplify the data a bit so it's easier to look at and compare to our flow cytometry data. Between these 2 different techniques, we did have a couple similarities. For example, with our SCOV3 EVs, we found that there remained the highest detection by CD81 between these 2 techniques. However, overall, we found that there was a lot of disagreement between the apparent protein profiles on these 2 different techniques. This wasn't what we had expected, but after thinking about this, we thought, well, maybe this is something that is due to the difference in limits of detection of these 2 techniques. So with ExoView, the limit of detection is based on how many fluorescent antibodies are on a single EV. So you need enough fluorescent antibodies to be identified above background. This means that there's no given size limit of detection, and we expect that ExoView sees a large portion of the total population of EVs. However, with flow cytometry, we use side scatter as our trigger. So we need EVs that scatter enough light to be detected by the flow cytometer. A bunch of different groups have sort of started looking at what this means for like the physical limit of detection for flow cytometers if we're using scattering, and using flow cytometers that are developed for cell use. Some labs have found that there is sort of a limit of detection between 70 and 80 nanometers. So after reading this, we looked at a sort of size histogram of our own EVs from transmission electron microscopy, and found that the vast majority of our EVs were below about 100 nanometers, but our mode size was somewhere between about 30 and 40 nanometers, which is much lower than this limit of detection that a bunch of different groups had suggested for flow cytometers. So looking at this, we wanted to see exactly what our limit of detection was for our specific flow cytometer. So we used a method that was developed, I believe, by Joshua Welsh, and I think Edwin Vanderpoel has also done this, where we analyzed the scatter off of a variety of refractive index standard beads. And using a bunch of different characteristics of our own flow cytometer, we were able to develop a model of the side scatter that would be correlate with the exact size of the EVs that we were analyzing. So doing this, we found that our limit of detection for our flow cytometer was only about 100 nanometers. This meant that we were only analyzing a very small fraction of the total EV population, whereas with ExoView, we think that we were able to see a much larger portion of our actual entire EV population. So we expect that the differences in these two different protein profiles was at least in part due to these differences in limits of detection. So it was unfortunate that we were not able to recapitulate that data between these two techniques. However, we believed with our ExoView data that we had seen enough evidence that there were these subpopulations to confirm that our SCOV3 EVs did have these different tetraspanin subpopulations. This brings us to our second question. Can EV capture by a single tetraspanin affect identification of ovarian cancer markers? So now that we had seen these subpopulations, was this subpopulation expression of clinical relevance? So far, we had been using the same sort of capture methods as many of these diagnostic tools, using single EV capture and comparing the three of them. However, to answer this question, we needed a way to non-specifically capture our EVs and get an idea of the entire population. Currently, there's no really well-accepted universal marker of all EVs. I know that there's a lot of work in this. Syntenin-1 is sort of a rising candidate. But we instead decided to use biotinylation of our EV population using this molecule. This molecule should non-specifically bind to free amines on protein surfaces. Since all EVs should have some form of protein on their surface, this should be a great way to non-specifically label the entire EV population. So to do this, we incubated our EVs with this biotin molecule, washed away any of the unbound biotin molecules with side exclusion chromatography, and then incubated our EVs on top of this capture chip that we had been using, now with the addition of an antibiotin capture spot. So this would be our non-specific capture spot that captured hopefully a sort of representative sample of our total EV population. And then we could compare it to the single tetraspanin capture spots and see if there was a difference. So before assuming that this was a great non-specific label and great non-specific capture method, we wanted to confirm that our biotinylation didn't affect the apparent tetraspanin profile of our EVs. What I mean by that is it's possible that there was some bias in which of the tetraspanins or which of the proteins that the biotin bound to, and that we would get sort of a lack of signal from these tetraspanins or proteins because of this. Since we were trying to see the general population, we didn't want this to happen. So we had to confirm that this was not the case. To do that, we used our same tetraspanin fluorescent antibodies and looked at the single EV capture spots to see if there was any difference between native EVs and our biotinylated EVs. So here you can see that overall, there was very little difference in the capture and detection of our EVs after biotinylation. There was a significant decrease in the CD9 capture CD81 detection, but overall it was a very low percentage. And this was the only combination capture detection of the 9 total combinations that actually had any significant difference. So this supported that we weren't getting any bias in our apparent tetraspanin profile from our biotinylation, which was great. Next, we started looking at our biotin capture spot. So this was our non-specific capture spot that should be grabbing EVs that were biotinylated. Here we found that compared to any of the single tetraspanin capture spots, we were actually detecting more EVs on this non-specific capture spot. This was really exciting. It meant that we may get increased sensitivity to our EVs when we did this non-specific capture method compared to using these single EVs. So before moving on to our ovarian cancer markers, we wanted to just check that this biotinylation worked for many different populations of EVs. So we did the same biotinylation on our serum and placental mesenchymal stem cell EVs that I had talked about previously. We found pretty much the same thing where we got about the same or more capture by our biotin capture spot compared to any of the single tetraspanin capture spots. But we also found something that we found was very interesting. And we call this multiplex bias. So what I mean by this is for our serum samples, for example, if you look at the CD81 capture spots, there is very low detection of CD9, very low detection of CD63, and high detection of CD81. However, when we look at our non-specific capture spots by biotin, there's very high detection of CD9, high detection of CD63, and low detection of CD81. This means that we're not getting an idea of the general population when we're looking at this single tetraspanin capture by CD81. It's not indicative of the entire population. We call this multiplex bias because it means that CD81 doesn't necessarily multiplex as much with the other ones, or at least we're not able to detect that. We also found this in our placental mesenchymal stem cells. For example, with CD63 capture, there's very high detection of CD63 compared to any of the other 2 tetraspanins, whereas in our non-specific capture, we found that there was very high detection by both CD63 and CD81. This meant that we were changing our sensitivities to other molecules when capturing by a single tetraspanin. And our question after this was, can we also see this with ovarian cancer markers? So are there specific pairs of tetraspanin and ovarian cancer marker that multiplex more than others? So to look at this, we replaced our fluorescent antibodies with fluorescent antibodies against common ovarian cancer markers. These were CD24, EpCAM, and HER2, all of which that are expressed on specifically our SCOV3 cell line and which have been identified on SCOV3 EVs. So the first thing that we noticed when we did this was that for any of these 3 different markers, there was one specific tetraspanin that seemed to have higher sensitivity to these markers than the others. So for example, with CD24, CD9 capture was the only way that we could actually see CD24 above background. This means that had we chosen, say, only CD63 to capture these EVs, it's likely that we would have had lower sensitivity or wouldn't have been able to see these CD24 positive EVs at all. For EpCAM and HER2, CD81 seemed to have the highest amount of multiplexing with these 2 different markers. I would like to note, for none of these 3 ovarian cancer markers was CD63 the most sensitive pair. And a lot of different diagnostic tools tend to use this CD63. So something to think about. The second thing that we looked at was our non-specific capture spot. So for each of these 3 different ovarian cancer markers, we found that this non-specific capture by biotin was able to see just as many or more of these ovarian cancer marker containing EVs. This was especially important for both EpCAM and HER2 detection, as we were able to see 5 to 10 times more EVs when we were doing this non-specific capture compared to any single tetraspanin. Since the main issue right now with ovarian cancer diagnostics is this sensitivity, this could be really important. Using this non-specific capture method may be a great way to get around the sensitivity issue and really increase our sensitivity and use these EVs to the best of their ability. So with that, I'd like to give you sort of 3 key takeaways for this paper. The first is that studies utilizing single tetraspanins to label or capture EVs may be examining only a subpopulation of EVs. So it's all right to use single EVs, but we also always need to remember that we are only looking at this subpopulation, and this may have these subpopulations may have differences in their multiplexing or for the field at large, difference in their functions. Since there's been little or very few studies on this so far, it's really hard to know what the effects are of looking at these subpopulations compared to the entire population as a whole. Second, uninformed single tetraspanin capture can bias results and decrease sensitivity. So as we showed here, which tetraspanin we chose to capture by affected how many of these ovarian cancer containing EVs we could identify. So this means that if we chose one at random, we could have chose one that had lower multiplexing or a lower amount of these multiplexed ovarian cancer biomarkers, and thus decreased our sensitivity to it, when our entire goal is having the highest sensitivity we can to these different markers. Finally, examining EVs at the single particle level provides clinically relevant information that bulk analysis does not. So looking at these different subpopulations, identifying which have good multiplexing efficiency is really important in the diagnostics field. But I think it's also very important to the field at large. Understanding these subpopulations and the importance of this heterogeneity is going to be really important in the next couple of years, I think, in the EV field, and it will allow us to use these EVs to the best of their ability. So with that, I would like to thank my lab, specifically my two mentors, Dr. Randy Carney and my co-mentor, Dr. Aijin Wong, as well as everybody who helped me on this paper, who is listed in red, and all of the funding sources for our lab. With that, I'd like to thank everybody for their attention and open up the floor to questions.
Thank you, Rachel, for this great presentation. That was very, very interesting. People were mentioning, I think you described pretty well, but just to make sure that everyone understands, there's some question regarding the fact that, for example, you were using CD9 as a capture antibody, and you were not necessarily able to detect CD9 with a detection antibody.
This is something that we saw very frequently, and that confused us as well. So on our CD9 capture spot on the bottom left, we saw very low amounts of CD9 detection. And this is probably a combination of a couple things. So it's number one, there are different sensitivities or different limits of detection in these three different fluorescent channels. So there's lots of autofluorescence in the blue channel. So it has to have a higher intensity for us to be able to actually see above background those EVs. So the blue channel is definitely the least sensitive. So we're definitely missing some EVs in that sense. But another thing that is probably sort of at play here is that there is some sort of steric hindrance. So we were talking about this a little bit, but when we are capturing by CD9, it's likely that there are other CD9 molecules that would have been able to be bound with these fluorescent antibodies, but that are now blocked because the EV itself is captured by that molecule. So it's likely that there are spatial differences, and since we're blocking that from being bound, it can no longer be sort of identified. So it's probably a combination of those two different things, but it's, yeah.
I'm jumping on actually one of the comment or questions from just come out, like from Tiago Costa-Coelho. Do you think that it's possible that there is some soluble form of tetraspanin that can be co-isolated with ultracentrifugation and actually compete for the binding site of the antibody?
It's possible. But I don't think that that's at least what's happening here. So we definitely find that if we do have soluble forms of things or soluble proteins, that we do get less capture. So that happened with our biotin. Initially, we didn't do that size exclusion chromatography to get rid of the protein. And we saw almost nothing captured on the biotin capture spot. But I don't think that that's the case exactly for CD9, at least, because when we did size exclusion chromatography, we saw the same exact thing happening, where it was very low detection of CD9 on the CD9 capture spot. So I think this is probably that CD9, just there aren't that many repeats of that exact tetraspanin on these SCOV3 EVs, so we're just not able to see it as much.
Could any other tetraspanin be used as EV markers except CD9, CD63, and CD81?
We haven't tested that. It's possible. I I think that the reason that these 3 different tetraspanins are used so commonly is because people were able to find them the most. But if there is a different tetraspanin that is frequently on the entire population of EVs, but at a lower rate, and that's why we haven't used it before, that would be a really great candidate for this primary capture, but I'm not aware of any at the moment.
Hi, actually I have one more data analysis question. Why you completely don't have the negative control signal in your graph from ExaView? It's
Yes.
quite unusual for, is it because of secondary analysis or is the raw data from ExaView?
So this is, secondary analysis. So the way I chose my sort of limit of detection to say, yes, this is an actual particle, was looking at our negative control, the mouse immunoglobulin control. And I said, I don't want to see more than about 10 total what the machine quantified as particles. So there are lots of things that it can identify as particles. It just depends on where you choose your limits of detection. So had I chosen a slightly lower limit of detection, we would have seen more of a peak in our negative control.
Question from Louis Vu. How do you explain the fluctuation of fluorescent signal at concentration lower than 500 million? So I think it's your previous slide on the,
So this is actually something we wondered about as well. This fluctuation is probably because we were getting fewer and fewer EVs that we were actually looking at. So at those lower concentrations, we were looking more in the hundreds of particles. So we didn't have as tight of an idea of what the actual population was. So that's why we got those different fluctuations, just because we weren't getting as great of a number total.
And also regarding the ExaView, William Ross was asking, do you have test, retest reliability data for ExaView?
Yes, when we do retests, it's very tight, different error bars. I don't think this one shows it here, but we did this week 1 3 times in a row, and it showed exactly the same thing. And interestingly, when we did those different 6 weeks, there were slight differences in the 6 weeks of those SCOV3 isolations, and those did stay the same. So there were slight differences in the subpopulations, and we could always recapitulate that. So the same isolation of EVs always looked exactly the same.
And to jump into another questions on so regarding the ovarian cancer markers on serum,
yes.
Have you tested if the different markers on serums, if there is any differences? Also, could you give suggestion on which point during cancer progression that we can start to detect the markers via EV's first markers? Yeah, do you think that there is some sort of like tumor burden or aggressiveness that you can have more sensitivity with the EVs and the surface markers? You can detect
that. That's a really great question. There probably is. Since we were using a cell line for this, that's not something that we specifically tested for. And SCOV3 is a pretty aggressive form of cancer, I think. So it's hard to know. We hope that this would be a great way to increase the sensitivity. But since we didn't have those serum samples from patients, say, in early stages, it's hard to know if they also produced enough EVs to be detected by this way. So we hope that non-specific capture could be a way to get higher sensitivity. Mm-hmm. But if we're not producing those EVs and seeing those markers enough, it possibly couldn't work the way we would hope.
And actually, I'm gonna combine a couple of questions that we have from John Sperduto. We actually cannot use his mic, and also Carlos Jesus. So with the ultra centrifugation that you have used, we can have co-isolations with some lipoproteins. You can also have, let's say, like some sort of impurity or affect the integrity of the EVs. Do you have any recommendations that to actually assess the integrity of those EVs and the presence of contaminants, how they can affect either your detection on the current platform, also by using the antibody biotin capture method?
Specifically, I think you're asking, so with the biotin, we will get biotinylation of the proteins as well. And that is totally true. I think I would recommend doing a a couple of different isolation techniques. So here we did ultracentrifugation initially, and then did the size exclusion chromatography to get rid of any of the free proteins that would also be biotinylated. And that really helps with not getting that background of those proteins that also would be captured. Integrity wise, I think the same thing is probably true. Doing a combination of different methods, it means that we're I guess isolating by different characteristics of the EVs. That helps us ensure that we do retain the integrity of our EVs. I know by differential ultracentrifugation that we do get sort of some changes because of those high forces. A thing that we could have done, I guess, would have been density gradient to confirm that we weren't affecting too terribly the integrity. But since we were able to capture and then detect by antibodies, so theoretically on two different sides of the same EV, I think that it's likely that we did have true EVs that had that vesicular structure still.
A question, so for the flow cytometric data, did you use the same fluorescent channel for the 3 different markers?
Yeah, so we used the same exact antibodies that we used with ExoView. So I believe it was Alexaflora 488, 555, and 647. So we used those same exact antibodies with the flow cytometry, and then used the different channels that we had available on the flow cytometer. Those are probably slightly different sort of bandpass filters than what we would see on the ExoView itself. So there is a possibility that since we were detecting, I guess, yeah, with different sensitivities of those exact markers between the two different techniques, that also could have caused some of the discrepancy that we saw.
Did you check if all antibodies show a similar affinity for the targets? I mean, I guess that's an important question that could be difficult to answer. Like if CD9, CD3, CD81 have different affinity, and if this different in affinity can actually result in a heterogeneity that you see on the detection.
Yeah, that's a really great point. I think that there probably are differences in the affinity for these different markers. So it's important to note that it's only a great quantitative comparison when we're looking at the same exact fluorescent channel on the different capture spots. I know that still is dependent on the antibody that is capturing them. So if there are differences in affinity there, it's not great. But since we saw these differences in the actual populations of EVs, so SCOV3 EVs versus our serum EVs, I think it shows that although there may be effects from those affinities, we still are able to detect significantly different populations. So it's not completely quantitative here. We can't say because we didn't see them, there isn't a subpopulation that has this multiplex pattern, but we can quantitatively compare the two and say there's more in this than there is in the SCOV3 EVs, for example.
Thank you, Rachel, and thank you. Yeah, I am satisfied. This is a very interesting point that eventually we need to research more in the next experiments and next research. But it's very, very interesting to take in account when we analyze this data.
Yes, yeah, I totally agree.
Thank you. Thanks so much. Thank you, Alexi. I'm looking if we have any other questions. I think there's a lot of good comments about your presentation. Great work, great presentation, amazing talk. So that's good news. I think you have a good reception from the manuscript. If anyone has any questions on the chat, please feel free to indicate them. So yeah, so I mean, if I take just a more open-ended question, so moving forward, what would be your strategy, even if you don't want to give the chef recipe, right, like in a restaurant, but what would be your suggestion if anyone wants to start using the ExoView system in terms of antibody to use for capture and detection?
Yeah, I mean, I think that the chips they sell with the 3 different tetraspanins are super useful for this because, yeah, most of your EVs seem to express at least one of the tetraspanins. Comparing and seeing which of those seem to multiplex the most with a given marker, I think is a great place to start for people that are developing these diagnostics. I would have loved to test different serum samples of these ovarian cancer patients and see if we could develop a biotinylation method that worked with that, since it is so much more complex. But yeah, we unfortunately didn't get to that point with this study.
Yeah, that's great. And I think, like Ken just put in a chat, there's a lesson here to have about our understanding of the biology of biomarkers. We were initially starting with a bona fide marker of EVs, but more and more we realize that it's more complex than that.
Yeah.
And you perfectly illustrate using the ExoView system supported by the flow cytometry that it's a complex mechanism, and how the cells express the tetraspanin, how they process, and also the technical approach that how they can affect also the readout. We have.
If I could I just make a point about that too? That I do think that it's interesting, the findings that you have on CD63 versus CD81, because we know that there's no universal marker of exosomes or microvesicles, ectosomes, but CD63 does tend to be more enriched in those exosome kind of fractions, whereas CD81 is more enriched in the plasma membrane origin vesicles. And so, we as a field, I think, have really emphasized the exosome to the exclusion of other types of vesicles. And what your data show is that really we can't do that. If we want to be sensitive to biomarkers, we have to be open to the possibility that there are vesicles that are released from the plasma membrane. And that makes a lot of sense, actually. If you're talking about some of these surface markers, we don't want to throw out the stuff that might be most informative for us. So I think this is a very nice contribution that you have here, reminding us of keeping the purpose of our biomarker research in mind, rather than trying to justify our presuppositions.
Yeah, exactly. Yeah.
And I think Ken, on that note, I think which is interesting is a question that I already heard people asking is, what's the correlation between the abundance on the cell surface and that you find on the EVs? Is that necessarily like we can think about like a passive mechanism, and if you have more expression on the cell surface, you necessarily see it on the EVs, but at what extent? This is also like very provocative. We have two questions actually. Artem was asking, have you tried to label the EV cargo with the ExoView and an image with the ExoView?
No, so I've seen that they have a new protocol for this, and I've never tried this, but I'm really interested in seeing. I think that could be really useful and make this technique a lot more useful for a lot more people in the field. So I'd love to hear if people had success with that. Yeah.
If I may, I'd like to ask Gabby Lima to come on, if you're there, Gabby, to just tell us a little bit about what you've done with internal cargo.
Yeah, I've tried a few things, testing the protocol, also synthenning. It seems that the protocol really works for synthenning, at least so far. We've also tried a few specific markers that we are interested. I do remember on the top of my head here seeing some of heterogeneities also when certain, like a cargo that you see in combination with a tetraspanin. So this is also something we can find on using a cargo marker too. Yeah, so the protocol is good. It works. It's also pretty simple too. It's pretty straightforward. And we're also still trying a few things here to see what we can come up with.
That's awesome to hear.
Thanks, Gabby. Thanks for sharing that.
And Lilia was asking, Lilia Panyushkina, could you use this tool for tetraspanin colocalization detection, like thinking of spatial conformation?
I think so. So we tried a couple of things to look at the spatial arrangement to see if it was that we were getting these patches of tetraspanins, and that's why we had that odd low detection of CD9 on the CD9 capture spot. And we did find that when you pre-labeled with CD9 antibody before allowing them to be captured on the chip, that we got very low capture of the CD9, as you would expect, since we had the antibodies all over that. But on the other different capture spots, so CD63 and CD81, we saw many, many more EVs that were able to be detected by CD9. So I think that that probably is an indicator that there is this spatial arrangement of them. I think it'd be cool to do sort of FRET on this system if you can get enough signal to be identified above background, and that would be a really cool way to look at the spatial arrangement, though it's not something that we ever really went after doing that blocking study.
And maybe one or two last questions. So Catherine Miar Askel.
So my question was about the presence of lipoprotein, since we know that serum has a bunch of it. And I know from my own experience that doing ultracentrifugation won't remove LDLs and less dense lipos. So how did you account for the contamination? And do you think it would influence your experiments?
Yeah, that's a great point. We did find, so not specifically, I guess, with lipoprotein, but we tried labeling with different proteins like CFSE, and that the presence of these, we did get some non-specific binding when there was a ton of this protein around. So I think if you do have things like serum that are these complex biofluid sources, you do have to do those two methods of isolation if you're doing the biotin capture. The antibody capture spots, they don't have a whole lot of non-specific sort of interactions with proteins for the most part. So we were able to see with our serum samples just from ultracentrifugation isolated samples that there was very little background compared to our different cell culture methods. So to me, that indicated, even though we do have those like low density lipoproteins and stuff in those isolates, that because we're using the specific capture by the tetraspanins, we really don't see much of that in this specific method. If we're using the non-specific capture, definitely that would be more of an issue, and we would have to pair a couple of different isolation techniques to make sure we got rid of most of the lipoprotein before it would be useful.
Before I let Ken to maybe has a message to say or something, but on the behalf of many of the attendees, congratulations to Rachel for the great presentation. That was very exciting. A lot of questions, and I think a lot of provocative data, like I said, to really like help us guide us through the use of those technology for EV measurement. And Randy, I mean, we expect to go on 3 years in a row and next year to get another paper with great data like that. We're gonna wish you the best. Ken, do you have anything to before closing the session?
Only to say thank you again. And thank you to you as well, Fabrice, for this very nice moderation today. So thank you everybody for joining, and we look forward to seeing you again next time. Be well. Take care, everyone.